hey guys, my son is not feeling so hot, so I might be out tomorrow, and I wanted to go over and do some notes with you real quick just so you're prepared for what's going to happen in class. So even if I'm in tomorrow, this is still a good review for you to watch at home. So last class we talked about DNA replication, and you took these notes. Pause here for a minute if you don't have your notebook out, and get your notebook out to review. So DNA replication is when we copy DNA. It's really important whenever a cell undergoes mitosis or cellular reproduction, right? So when you get new cells, you need all new cells to have DNA in them. And that's in the nucleus. So the DNA has to copy its DNA for those new cells, okay? There's 3 steps we talked about. First, an enzyme comes along. It's called DNA helicase and unzips or opens up the DNA. Then, number 2, free-floating nucleotides attach to form 2 new complementary strands. And step 3, the entire molecule unzips and is copied, and it makes 2 what are called daughter strands, okay? And we looked at some video of that in the Hank Green video posted on Google Classroom. Will help you review that too. Okay, now, sometimes that can go wrong, and we can get mistakes. It's not often, but it does happen. And it's actually a beautiful thing because that's what leads to evolution, baby. So difference in genes, and that's why we have blue-eyed people and brown-eyed people now, because of these beautiful mutations. Okay, now, mutations aren't always bad. Like blue eyes aren't a bad thing. They're a little more light sensitive, but they're very pretty and cause people to have more mates, and so that led to evolution. Some chemicals and UV light can cause mutations. Some are bad and lead to cancer or freakishly weird diseases. But some mutations are good and lead to evolution. And some mutations are like neutral and they don't do anything. Like birthmarks. That's a skin pigment mutation. It's not going to kill you and it's not going to lead you to evolve better or reproduce more. But it's like a neutral thing. It doesn't hurt or harm you. Okay? So today we're going to talk about making a protein. Fancy called protein synthesis. So proteins are the most important components of all living things. Like you need proteins to survive. And they perform a wide range of functions in your body, and you have to make a lot of them. They make up your hair, your skin, your fingernails, and control your bodily functions. So enzymes, natural steroids that are in your body all the time, your hormones, those are all proteins. So when you look in the mirror, you're looking at a really nicely packaged together set of proteins. Look at how handsome you are. Oh, you protein sack, you're beautiful. Okay? And DNA holds the code for making those proteins. It's like the directions for making you, or the recipe for you. Okay? So, here's the problem though. DNA is in the nucleus, right? It's all the stringy stuff called chromatin, and it's surrounded by this nuclear membrane. Like it can't get out, because if it got out, it could get damaged, and that's your recipe book. And if that got damaged, you'd be making the wrong proteins, and it would be a hot mess. So it has to be protected in this like sealed vault of the nucleus. So that's a problem, because if it has the code for making proteins, and proteins, if you remember, are made in these little dots. What are they? Shout it out. Ribosomes. Good. Okay. So those are ribosomes. So how does this code get to the ribosomes? Well, that's the key, right? There has to be something that can go in, read the code, and go out and tell the ribosomes what to do. And that is going to be really important. So again, where are proteins made? Ribosomes. Where is DNA? Nucleus. So how does the code get to the ribosomes to make those proteins? Messenger RNA, baby. RNA is a molecule that's very similar to DNA, but it has 4 differences. You have to know these. I'm going to post this slideshow to Classroom so you can take notes after, and you should on this stuff, because I am zooming through this so that the sub doesn't have to sit there forever and listen to me, or so that you can pause whenever you want to at home and redo this. So RNA has ribose as the sugar. DNA has deoxyribose, right? RNA is single-stranded, one strand, not a double helix, a single. RNA has a new nitrogen base called uracil. And RNA can leave the nucleus. DNA can't, remember, because it could get damaged. It's got to be protected in that safe little nugget, or the safe little nuclear membrane. So again, where DNA has deoxyribose, RNA has ribose. And where DNA has A, T, C, or G, RNA has A, G, C, or U here. There's the difference with the little circles next to them. Okay, so here's DNA double helix, here's RNA single helix, and again, CGAU instead of CGAT, okay? And again, the sugar is deoxyribose, not shown here. Okay, so mRNA will go into the nucleus of the cell and copy the DNA. That's called transcription. When you write something down, like when you copy these notes, you are transcribing them into your notebook. You're putting them into your own handwriting. So mRNA goes in and reads and copies the code, but uses uracil instead, so it's like writing in its own handwriting. Okay? That mRNA then goes to the ribosomes to make the protein, and that's called translation. This is a simplified form of all this, but for our purposes, it's enough for today. Okay, so if we have a DNA sequence that's ATGC, what would the mRNA be? Well, I know that A binds with U in RNA. T still binds with A. Oh, excuse my terrible font. G still binds with C. C binds with G. A binds with U. T binds with A. G binds with C. C binds with G still. And C binds with G always. Those never change, so that's great. Okay? So now this is, I know it's ugly looking, but you get the idea. That's a U, not a V. This is what the RNA would look like. Okay, try one without me. So if this is the DNA sequence, what would be the mRNA sequence? Pause if you don't know. T binds with A binds with U, so it would cross, the RNA would be A, U, G, G, U, A, G, U, A. Okay, we're going to skip that part. Transfer RNA is next. Once the mRNA gets the code, it goes to the ribosome to make a protein, and that's called translation, right? Well, to review, transcription is when DNA goes to mRNA, mRNA copies the DNA code, and translation is when the mRNA turns it into a protein. tRNA, or transfer RNA, is used in this process. You're going to make a manipulative in just a moment, and in that you're going to have to fill in the appropriate boxes to make as a homework grade. Let's just put it that way. So I want to review with you one more time, okay? Proteins are so super important, right? They are the most important components of living things. Their creation or synthesis is important for cellular function. Proteins are made of amino acids, and our DNA provides the instructions for assembling amino acids in their proper order, thus creating proteins that can do their jobs correctly. The nucleus holds the key for protein synthesis, a process that takes place within every cell of every organism or living thing. Okay. In the cell's nucleus, DNA is used as a template and copied to create messenger RNA during a process called transcription. mRNA is similar to DNA, the double helix, but it is made of ribose instead of deoxyribose. It's single-stranded, and the nucleotide-based thymine is replaced with uracil. mRNA then leaves the nucleus, travels into the cytoplasm to begin translating the mRNA sequence into the protein. Ribosomal RNA, which I didn't really talk about in this presentation, but ribosomal RNA is called rRNA, is made up of a group of proteins which form ribosomes. When mRNA leaves the nucleus, it locates a ribosome in order to begin building proteins. The ribosome moves along the mRNA strand and reads the sequence in order to translate it into an amino acid sequence, which is what proteins are made of, right? During translation, the ribosome reads 3 mRNA nucleotides at a time with a nucleotide triplet, which is called a codon, calling for a specific amino acid. That is where tRNA, or transfer RNA, comes in. As the ribosome reads the nucleotide triplet or codon of mRNA, a specific tRNA molecule matches up with the sequence and carries with it a specific amino acid. The amino acid then binds to the growing amino acid polypeptide chain until the mRNA message comes to a stop and the protein is created. That, my friends, is the whole process. So what I want you to do now is remember that you saw this video and know that you can always go back to it to find the answers in today's manipulative. What you're going to be doing is cutting and pasting into your notebook. I said a lot all at once, and I went very fast at the end. So if you need to review, you can pause and rewatch at any time because this is posted to Google Classroom and my YouTube page. Okay? You got this. Be good.